This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
This is Paul Verschure together with Tony Prescott for the Convergent Science Network podcast here at the 10th edition of the Barcelona Cognition, Brain and Technology Summer School. And we're here with Ton Coolen. And Ton, in your talk this morning, you were dealing with the analogy or the similarities you might find between neural networks and immune networks.
Yeah.
So how did you come to study or to raise that specific question? What kind of mileage do you think we can get in addressing it?
Yeah, how did I come to it? It was really a case of being lucky to be asked by a group of Italian researchers who had recently developed some really nice new models. And in the development of these models, they find that at some point, solving these models required mathematical technology, which had only been in the public domain for a couple of years, and they were not having the experience to do this calculation, and I happened to have been working on similar problems. And in addition, we knew each other because one of these Italians had done a PhD in London, so one thing led to another, and I was asked to join.
Mm-hmm.
And I really enjoyed that. It was really very interesting. The model was interesting, and it allowed me to do research in a field that was really very close mathematically to the borderline of what was then the state of the art. So this problem had everything you would wish for in a problem. It has clear relevance. Understanding the immune system has clear medical relevance. And it also was very, very interesting scientifically, as someone who likes to do a calculation, to be involved in those calculations.
So what were the specific mathematical techniques that they needed?
Yeah, in the analysis of systems with very large numbers of variables, the traditional methods that were being used until around 2000 work only when the number of components that each element interacts with is either very large or very small. But in the intermediate regime, where you have components that interact with a finite number of other parts, there were no methods available, and this is called finite connectivity analysis, and there are different mathematical tricks for dealing with it. But they'd only been developed in this domain around 2000 by various people, including mainly Rémy Monasson in France, Giorgio Parisi in Rome. And this what turned out to be needed to solve these particular immune models.
And these techniques were already developed looking at specific natural phenomena, or these were being developed within theoretical physics or applied mathematics, just as a technique that people thought was important?
Well, these methods tend to come from a community of solid-state physicists, theoretical physicists, but it is specifically the subpart of that community that look at interdisciplinary problems. So I think the motivation for looking at finite connectivity problems might have even come from optimization problems in computer science that were being analyzed by methods from theoretical physics. At least the names that come to mind at this moment are typical type of people who've been working on optimization problems. They convert these optimization problems into theoretical physics problems and then use the technology.
So what I find instructive about that is that what you see here is how applied problems are driving the basic science.
Oh, yes.
Right? Which
That's
is actually,
very much so.
It's counterintuitive. Usually there's this idea like, okay, the basic science happens because we have this genius sitting somewhere in a dungeon somewhere, and then it comes out and becomes applied. But here you see it exactly the other way around.
Yeah, I mean, this has been going on for about 25, 30 years at least in that community. Initially, physicists started looking at heterogeneous systems in the context of some obscure magnetic materials that later turned out to have no use whatsoever anywhere. They were called spin glasses. And since then, the main application domains of the mathematical methods that came from these obscure materials have been ranging from computer science to economics to biology to medicine to statistics. And now there is a very fruitful flowing back and forth of ideas and problems between theoretical physicists who are only theoretical physicists by virtue of the salary they draw from the physics department, but they work largely on non-physical problems. So this has gone on for quite some time.
So one thing I found really remarkable in the model you presented from these people in Rome on the immune system in this case, is that they actually stumbled, if you want, into a description of this immune system in terms of an energy landscape, if you want, or a dynamical landscape that you could call energy, that was identical to what people like John Hopfield have proposed now 30 years ago as effective macroscopic descriptions of the spin glass models initially and then of neural networks. So is this convergence, was this happenstance, luck, bias because of the techniques people had? How did that come together and why is it meaningful?
Well, I think it's a mixture of many things. First of all, you have to have seen certain domains to recognize certain approaches or models or methods for what they are. So it demands that you are someone who is willing and able to look over the fence away from their own discipline. And the people who did this in Rome had been involved in various projects related from physical systems to neural networks and so on. So while doing the calculations, while building the models, at some point you recognize that models are developing into a certain form that resembles something else. But you will only recognize it if you've actually been reading papers or preferably been carrying out research in these other disciplines. So breadth helps you. And also, the other thing you mentioned is mathematical technology. Mathematical technology constrains what you will be writing down in the first place because you know only certain types of models will be solvable. So you steer away from constructions that you know will lead you into the morass anyway. So all these things work together.
So from your talk, I understood that both the neural networks and the immune networks have this beautiful abstract description in terms of statistical mechanics, which you've explored in the talk. But I'm interested in, I think you also pointed out in the talk to some other sort of direct analogies between immune networks and neural networks that could provide further levels of interaction between those two fields. Can you say a bit more about what you think those are?
So the similarities are at different levels. You have the mathematical level that was already mentioned just now, but also even if you didn't buy these particular models, if you thought, well, this is just a choice by a couple of researchers and we could have written models in very different ways, although qualitatively perhaps similar. But at a higher level, you see that you are looking at two systems which share various fundamental ingredients. A, they have large numbers of variables. B, these variables interact with each other in a heterogeneous manner. And C, this system is not a static system. The forces that all these objects exert onto each other are evolving over time on the basis of experience in response to the environment that they work in. And these are ingredients that are exactly the same in neural networks and in the immune model. There are also differences, that's very clear. But the way you can exploit these similarities is first in terms of intuition. If you have, as is the case here, one field that has already been studied for decades, like neural networks, you develop a pretty good intuition for what to expect, pretty good intuition for what would happen if you added certain ingredients or took certain ingredients away. And that intuition is probably the most powerful thing to carry over to other fields, because the details may differ, but statistical mechanics teaches us that as soon as you have a very large system, microscopic details of how exactly the model is built or what exactly you choose for the types of variables or the parameters become less and less important. So when at some macroscopic level of description, what is built into the model is similar, then you also expect the behavior to be really similar. And that helps you. That helps you in designing models. And there is never a perfect model. Every model has a typical shelf life. But it helps you in being effective in selecting models, in selecting directions in which to push your models. Because at the end of the day, certain models are enormously powerful and helpful, and many others will not be so for various reasons. And this guides you, this analogy guides you.
And I think so for some of the neuroscientists in the audience, it was also surprising to hear you talking about immune networks and the way that we talk about neural networks as structures that have learning and memory, perhaps in quite a different way. And in some senses, the mechanisms may be similar and other ones might be different. But I guess that's one of the paths via which this research in immune networks might come back to influence the neuroscience.
Yes, that is something that is not guaranteed to happen, but it is something that I certainly think is worth thinking about. I think in science generally, it's very rare for one to make an interesting discovery, for that discovery not to have implications outside of the initial domain where the discovery was made. And the main short-term lesson that I think neuroscience can take from this immunological way of working is the possibility of having attractor neural networks in which one is not recalling one attractor at a time, an attractor which contains a very large number of bits of information, one image at a time, but where you see that there are multiple attractors which can be recalled independently at the same time, even though you are looking at a system that is fully interacting, not directly, but there are no subsystems that are disconnected from each other. But if you then change parameters, all of a sudden you can get a transition where all these subsystems are no longer acting independently. Now, many of the things that you think about in neuroscience are somewhat reminiscent of that, where, especially in making recognition on the basis of recognizing subparts of images, whether or not you combine them, or whether you want to create bigger objects that are composed of things that you've seen before, but never in that combination, may be relevant, may not be relevant, but certainly worth exploring.
Mm-hmm.
These models operate in a slightly different regime in terms of parameters and scales and numbers of bits of patterns and things like that.
But one thing I find interesting here is also how you describe the development of these techniques, right? We start with spin glasses. Those techniques made certain assumptions about symmetry and homogeneity of the system. So those techniques would not scale to complex biological phenomena like the immune system or nervous systems. For that, you need new techniques. Would that actually help us to also distinguish living from non-living systems? Because apparently, mathematically, there's a unique set of tools you need to describe living systems as opposed to a pure physical system like a spin glass. Would you go that far?
Yeah, well, one thing I've learned over the years working on biological systems is that when it comes to modeling, biology is much harder than physics. And there are some really very clear reasons for that. One very simple reason is that very often in biology, you look at systems in which the number of elements involved is neither really small nor really big. So the mesoscopic area. And the problem with statistical methods from physics is they only work when you have really large numbers. In biology, that's not always the case. That's the first obvious thing. The second thing is that many physical methods rely on the systems that study going to equilibrium. Well, that's never happening in biology, at least not until we are permanently horizontal. So that rules out about 3/4 of the methodology of statistical physics. But I think the main one, and that is also the most interesting one for a modeler like me, is that in biology, we always have heterogeneity. We have heterogeneity in interactions between elements, heterogeneity in molecular structures. And physics started out, at least statistical mechanics, started out looking at systems in which every particle was interchangeable with every other particle. 2 electrons are exactly the same. The forces between particles would always be the same. Physicists tended to look at pure systems, and they saw heterogeneous systems as, say, impurities of pure systems, perturbations of pure systems. Biology is not like that. Now, in the 70s, there was a whole area opened up in statistical mechanics to look at heterogeneous systems. This is where the spin glasses came in. But one fundamental difference has remained, and that is, in using the physical methods, you always have to be able to represent the heterogeneity as randomness. You write a probability distribution for the particular parameters or forces that are heterogeneous. And that's exactly what is different in biology. Biological systems are heterogeneous, but the heterogeneity is never random. It has always been a consequence, at least partly, of evolution and selection. And that rules out immediately all these methods. And the best example to think about it is probably the modeling of proteins. Yeah? Proteins are these large molecules assembled of little parts that are called amino acids. And nature uses proteins as the building blocks of cells, as messengers, and it's the basic hardware of cellular biology. If you look at randomly selected macromolecules of amino acids, they behave completely differently from proteins. That means that it is essential that in order to understand how these biological systems operate, you must study the evolution that gave rise to these structures. But that's a very complicated thing, because it is a two timescale process in which the processes on the slowest timescale, evolution, influence the fast ones, and the fast ones feed back to evolution. And you will never be able to write the heterogeneity in terms of a probability distribution. And that is really interesting, because it means that here is a type of problem that we cannot handle the way we would handle a heterogeneous physical system. And there are ways, there are openings, but it is a completely different field.
You mentioned this in your talk, that you were continually discussing with the biologists about which aspects of the system they considered to be important. And in your models, you have a noise term, which I guess at the moment captures a lot of that detail.
Yeah.
And there may be aspects of that detail that they think has to be pulled out and put into the model. Is what you're saying that the mass gets hairier and hairier as you pull more of those details out and put it into the model? Or is there some sort of possibility of actually going into that space and it's not going to become increasingly difficult? There are going to be ways of making models that are more biologically rich, sort of things that you can work with. You can still show interesting results.
I think there's no golden and perfect answer to this. It's always the case that in the development of models, you have these three distinct stages as I see them. So stage one is where you have a problem from the real world and you have really no idea how to model it in such a way that you can make progress. Now I think we are beyond that point in immunology now. And the second stage is that there are some models that seem to make sense, but they are really very simplified, extremely abstract, and one has sacrificed a lot of the realism in order to get something that works. That's where we are now. Then the third stage is, as you say, where you make these models more realistic. And that means having a good intuition for knowing in which order to bring these ingredients in. And there will be some ingredients that have a serious impact on the methodology that you will need to still solve the models. And there are some ingredients that are relatively easily incorporated. To give an example, in these immune models, the initial models of Adriano and Elena had the unwelcome element that a T cell, a single T clone, could be simultaneously excitatory to some B cell and inhibitory to another. It's just like, and that's a nice analogy, the same thing was assumed in neural network theory in the very beginning by Hopfield. We know that the hardware is different. The hardware doesn't allow for that. And in neural networks, we could remove this and we could still solve models. The same happened here. So that happened to be a relatively easy ingredient to bring in. There are other things that are going to be much harder. We can see that coming. And that is when you include the evolution of antigen, because then you have to study in great detail the dynamical processes of hypermutation, because it is a rat race between the virus or the bacterium that has come in and that will multiply and the immune system trying to catch up. So when you start working on those models, you can forget about equilibrium studies, you can forget about energy functions, because these systems are no longer going to equilibrium.
So you're going to move away from statistical mechanics to another modeling approach?
Well, one would move away from equilibrium statistical mechanics, right, which is the part that has been used so far on these particular models, and you would use non-equilibrium statistical mechanics, and that is a slightly more specialist area of statistical mechanics. It is a small subset of the broader area. Most of statistical mechanics is equilibrium.
But this non-equilibrium statistical mechanics has been developing by looking at biology, or it came from other domains of physics?
Oh, it came also from standard domains of physics, because even if you know what the equilibrium behavior is, you might still be interested in studying how the system would get there.
Mm-hmm.
So they were developed already within the physics community, but you just find that in biology, that will turn out to be your only toolbox, because most biological systems don't go to equilibrium.
But not so. Okay, the analogy between neural systems, biological neural systems, and immune systems is that they can learn and have memory, right? So in the nervous system, there's also many talks on this. The dominant view on that is that this memory has something to do with the connectivity in that network and how it's regulated. It might not be literally the synapse, but it is the regulation of the synapse that's key for memory. How should we think about memory now in this immune system? What's the substrate of memory in the immune system?
Yeah, that has been a very tricky discussion in the biology community, in the sense that there are various competing views. Some are fashionable in certain eras and then get ditched a couple of years later, and that has happened in this community. For instance, Jern's model of idiotypic networks envisaged a mechanism of memory that was very different from what most biologists now assume is the case. Most biologists now assume that B clones, after they've been activated, after they've been producing antibodies, then go into a state where they become so-called memory cells. So somehow you want to retain and memorize the particular shapes of antibodies that were very successful in attacking the antigen that came in. So the thinking is that those B cells that produce those antibodies, they convert into a state where they can live for 25 years. I find that intuitively slightly, not maybe unrealistic, but slightly far-fetched.
Is there any empirical support for that idea?
Well, I'm not an immune biologist, and I'm sure there is some support, but I would be surprised if that was a clear-cut and completely closed case. I think there is still room for.
But that would then predict that there is some reservoir of these memory B cells that reside somewhere in the body.
Yes, that's the problem.
That's expanding, right? That's completely expanding.
Well, the thinking is they don't expand. The thinking is that you have this group of B cells that have been activated, and they go and sit somewhere and they live 30 years, just in case at some point that particular virus reappears. I don't buy that, but I cannot argue that there is evidence to reject this. It just seems a bit far-fetched to me.
Okay, but it means for you, this means the memory problem for immune systems is still not resolved. We don't ever actually really know.
I think there will be an explanation which is a bit more natural and elegant than the memory cell.
So your modeling approach, would that lend itself to building models that can test the alternative hypothesis? So
I think so. Yes, I think so. But at this moment, we're not there yet, because the models are relatively primitive. I mean, compared to, if you look at that, that's why I find the history of neural network theory so interesting, because we've gone through many of those things, and you can look back and see, yeah, it was very similar then. Hopfield models were primitive, really very primitive. Lots was wrong with them. And the interesting thing was, at that time even, Hopfield models were still solved using equilibrium statistical mechanics, and they were, in these models, one sacrificed biological realism in order to be able to do that. And when you talked to people in those days, it was very often suggested, well, we will never be able to do the dynamics. So this is why we do it that way. Turns out to be not true. People are sometimes too pessimistic about what is possible. And that is very dangerous, because you miss opportunity. Turned out to be nonsense. Doing the dynamics was, in retrospect, relatively easy. Which was very good, because then we could remove all these biologically unrealistic model ingredients. I expect the same to happen here.
But now the other thing is that so for memory, it's mysterious. In some sense, we could argue memory is equally mysterious for the brain, but there is consensus on what that substrate would be, more or less. But now let's look at the learning component, right? So for the immune system, you would have hypermutation and selection upon this set of hypermutated B cells as your core learning mechanism.
Yeah.
Right? And Now, in the brain, yes, people like Jerry Edelman have proposed that similar mechanisms would work in the brain to select synapses and in that way have specificity of connectivity and learning. But it's not the only way in which we can think about learning in the nervous system. It might also just be the differential strengthening of synapses that allow you, as in a Hopfield network, right, to sort of bias the network towards certain responses away from other responses without necessarily selecting across a repertoire. You really remove certain possibilities completely. So do you believe that selection is then the only mode of learning in the immune network, or is it more subtle than that?
It's an interesting question. I think that over the years, we find that a lot more is going on than we presently think. I would also, for instance, expect that at some point there is some interface between the nervous system and the immune system. And even today, I had some very nice discussion with some of the people here, where these ideas came up too. If the nervous system, for instance, would be able to predict or anticipate certain invasions of antigen, then you can prepare the immune system, right? I think there's a lot going to be found that we don't know at present.
Well, in that sense, for instance, if you look at sepsis as an inflammatory problem, it's extremely interesting to look at. If you look at these broad organ systems, now they're affected by in sepsis by inflammation. It's a rather complex dynamics in which, let's say, the inflammatory load to organs can be very specifically or changing or transiently changing during such a disease process. And actually also many patients who suffer from sepsis, some of them might appear in all standard biomarkers healthy. They're sent home and a few months later they still die. Mm-hmm. Right? So that would also suggest there's much more active regulation going on of the global response. Yeah. Right? With the possible anticipatory component to the immune response that might or might not reside in the immune system itself. So how do you see that coupling then exactly between the brain and the immune system? Do you expect the brain to really model, let's say, the health status of organ systems and then bias in some way the immune response? And if so, what's the substrate of that?
You have to think really about these things in terms of the coding that the different systems use. The brains think in terms of electric potentials and the immune system thinks in terms of chemistry. Right? It communicates by docking and sending cytokines and things like that. And there are certain constraints if you think about possible mechanisms and possible interfaces. For instance, immune cells cannot pass the blood-brain barrier, so they cannot directly enter the brain. However, various molecules that they produce could, right? We also know that in the brain, as I understand it, there is an interface between communication by spikes and chemistry. The release of all kinds of chemicals happens also in the brain. So I don't think it's far-fetched. And there are all kinds of opportunities, but we will never be able to find out until we have at least a satisfactory description of the immune system itself. Mm-hmm. There's so much we still don't know about the immune system. Going to an interface now is premature. Mm-hmm. But if I were a betting person, I would put my money on there being one. Mm. It just seems to be giving you an enormous evolutionary advantage. And therefore, I think it would be there.
Right.
So the algorithms that are involved in learning, presumably, I mean, you didn't talk about them in any detail in the talk, but there's something analogous happening there to natural selection. And is it involving both the B cell and the T cell populations in some interesting ways that alters the population dynamics so as to give you some form of optimal service?
Yeah, so it's interesting if you look at the literature on immunology that most studies that have been carried out tend to have been carried out on B cells and on evolution of receptors of B cells. So a lot is known about them. Much less is known about T cells. And partially this is due to the fact that T cells were only discovered later. The regulatory T cells were only discovered in the 1990s. So there is a good reason why we know much more about B cells than we know about T cells. When you think about learning, and this is again where I find these models very helpful the models that create this mapping between neural network and the immune system, if you look at those models, you see where is the learning residing? What is the substrate for the learning? It is in the cytokine variable. So if you think about what they do, the cytokine variables simply tell you exactly which B clone talks to which T clone, and within these models, it is also in the other direction. And that is exactly what is being changed as a result of hypermutation and selection when we go through that process. Because if we change the receptor properties of the Bs and the Ts, then we also change who talks to whom. So we're effectively changing the patterns that, in the language of neural networks, would be the patterns stored in a Hopfield model.
So we're rewiring the network.
We are rewiring the network.
At the same time as we're changing the specificity of the cells.
So in a way, the network is a consequence of the information being stored. And since we change the information being stored, we're effectively rewiring the network.
So we could see the cytokine as a virtual synapse now that connects
No, not the synapse. The synapse is expressed in terms of the cytokines. So the cytokines would be...
They're the connection. They form the connection with the Bs and the Ts.
They are the equivalent thing of the bits in the patterns of the Hopfield model.
Exactly.
And they build the synapse.
So what's now the capacity of then such a memory? If the cytokines would then, in this, the analogy we're now pursuing, the cytokines are in that your connectivity matrix.
Yeah.
Right? So given the scale of the immune system and the number of elements, what kind of capacity would we then look at in these cytokines, and what kind of learning rates would you expect, or the rate of change that you can support?
Yeah, this we can now answer as a result of the models. And the answer is, in fact, very trivial. This is always when you have understood something, you know that you could have thought about it before. But in standard Hopfield models, what you know is that you can store, if you want to have perfect recall, only a relatively small number of patterns, but each of these patterns has an extensive number of bits. Here it's the other way around. You can store and recall an extensive number of patterns proportional to the number of B clones, but each of them will have only a finite number of bits of information in them. So the capacity is in the order of n, and precise numbers you find by calculating the phase diagrams, which has been possible now in compute these days.
Right. But then another kind of connectivity matrix might be defined by the T helper cells, for instance, that would modulate the interaction with the Ts and the Bs. Are there other, like for the nervous system, to just focus the whole learning problem towards the synapses keeps things relatively controllable and understandable. But maybe in the immune system, there might be multiple levels at which this plasticity now gets expressed, or multiple substrates. Or not? Are you happy with just focusing it fully for now at this cytokine modulation?
I think for now, this is what is being done simply because we are already very happy with what we can achieve with these models. But in the future, we will want to study in more detail the actual process of what you would say learning in neural networks, what we would call hypermutation, and thereby the changing of all these cytokine variables in the immune system. And then we will undoubtedly find that things get more complicated. We will have to look at different types of B cells, different types of T cells, and all those things. So you will inevitably get a next layer of complexity simply because you move away from what is currently one of the most primitive starting points imaginable. So that will happen. That will happen. But I think on top of that, we will find, and that is going to be done by experimentalists, we will probably find new players that we haven't even been thinking of. Not just B cells or T cells, but other cells that do things. We might find interactions, mechanisms that we don't know now.
But nothing about the signaling. So in the nervous system, in the end, it's electrical, as we discussed. In the immune system, it could be through the different metabolites, molecules that are going through the circulatory systems. What are the forms of signaling that now the immune system has available that would make it different from the kind of signaling you would see in the nervous system?
Let me think about that one. So, in the nervous system, I think there's one complication that maybe is not so much a complication in the immune system, and that is in the nervous system, we also have topology. We have wiring. And at least in the immune system, we don't have that because these cells just move around in the blood. The assumption is that there are no barriers that single out certain B cells as having given access to certain areas as opposed to others. There are some aspects only which are that the hypersomatic mutation takes place in certain parts of the body. But apart from that, we have no topology, and that makes your life a lot easier. So in terms of the signaling, there is the extra constraint in neural network systems that you have to look carefully at the wiring. These things cannot float around. Neurons cannot float around and establish and interact with whoever they want. And space is a tricky thing. And especially if you think about mathematical methods from physics. In one area, and that is space, they are remarkably unsuccessful. And that is when you look, for instance, at a very simple cubic lattice of molecules that only talk to their neighbors. That is still an unsolved problem. Even if these variables are just binary, they can be on or off, and they talk to their neighbors in a three-dimensional arrangement. It's an unsolved problem. So space brings an extra dimension that makes problems really significantly harder.
Mm-hmm.
And that is absent in the immune system. So that is a difference which actually works to the advantage of modeling in the immune system. And in brains, we can get away with it. We have been able to get away with it for a long time because at least neurons talk to a great number of other neurons. If neurons had only been talking to neighboring neurons, we would be in real trouble. And thank God we're not.
Right. So then, but how can we now test these models? Because also look at the case of neuroscience, right? There was initially also a lot of enthusiasm for the Hopfield kind of networks because people had this hope, like, okay, now it becomes mathematically tractable, neuroscience can become like physics, we can have analytic solutions, God knows what. But that was 85, more or less, right? So we're talking about 30 years. And has that impact been so overwhelming? Have we radically changed the way in which you think about the brain? Not so sure about that, right? So, on the other hand, it has been as a heuristic, it has helped people to pose certain questions. So now, in some, you can argue for the immune system, you stand now at the beginning of an equivalent process, right? Where this fundamental insight using these techniques from theoretical physics has helped you to get a handle on a system that actually you don't understand. But what's the trajectory that you now envision? What are the next steps to also make it empirically testable?
Yeah, so we are very aware of the danger of the same thing happening in immune modeling that what happened in neural networks, and I can come back to what I think are the reasons for that, because some of them are not scientific but psychological. Now, at the moment, what we're trying to do is we're trying to, in a particular multidisciplinary project that we've started in the UK, involving not just people like me who do models, but also bioinformaticians and experimental immunologists, we're trying to find indeed models in which we can test the predictions of the model using experiments. So we would do this in the form of having a model in which an antigen comes in that has never been seen before, and then predicting the dynamical response, first and secondary response, and see to what extent this is similar to what we get in what is called unchallenged individuals in an immunological response. And then try to build the models from there, test every time that we can make a prediction. At the end of the day, the only real test of whether you have extracted knowledge in any area of modeling is whether you can predict unseen situations with the model and then test them. If you can't, you haven't learned anything.
Mm-hmm.
If you can, you have.
And what's the status of the sort of the measuring techniques that the immunologists have to detect the sort of for your models, you're looking for quite a lot of detail in terms of the temporal and spatial dynamics, or certainly temporal dynamics of what's unfolding.
Yeah. So this is really difficult in the sense that it is very hard, and we do struggle with that, to find people with the right type of data, because you're looking for individuals who've never experienced a challenge by a particular antigen before. So you have to go for really rare diseases. You cannot go for flus or things like that, because there is almost no one who will have a primary response to that. So you have to go for very funny things like yellow fever. And our collaborators are trying to get these people from populations of individuals who are about to make a trip abroad to an area where they've never been and who are then being inoculated beforehand. And these are the ones that we try to get the data from, because then they get a response to this inoculation that should be very similar to primary response. Because then at least we can do tests before having modeled the secondary response, before having modeled the consequences of the hypermutation.
But why no animal models?
Well, why no animal models? It just happens to be that the collaborators we have don't work with animals. They work with people because they come from a clinical environment where it's animal immunology, human immunology they work on.
Right. And can you do some of this in a sort of petri dish where you just put some antigen into a little sample of blood? Or?
I've been told that this is really hard.
Right. Okay.
that it is one of the tricky aspects of experimental immunology.
But so look, here we have all these Brits. They're going on holidays to faraway exotic places where they're gonna run the risk to catch all sort of strange diseases, and they get inoculated. And imagine this whole thing now works. What's the specific prediction you would like to see tested first?
We would like to see the evolution in time of the distribution of clone sizes. Yeah. So if immune system is not challenged, so let's say before these people get the inoculation, the theory can do a prediction already on the distribution of clone sizes in normal state. Then comes a challenge. This challenge has a consequence, and you see this in the distribution of clone sizes. And the distribution of clone sizes is the typical thing that can easily be measured. But this is the first test. It doesn't mean that this is the perfect test. It just means this is a very clear, transparent test. And if it doesn't even pass that test, then you know you have missed something important in your model. You go back to the drawing board and you think what that could be. But this is relatively accessible data. And this is what we're focusing on. So it's a distribution of the sizes of the clones.
But it's like fine-tuning the model. Is the model falsifiable? So what would be the observation in this experiment that would completely kill the model that you now proposed with your colleagues?
Well, I don't know what the definition of completely kill is, because you can always go back to the drawing board.
Well, that's the question, right? Yeah, exactly. So,
but this is a falsifiable model in the sense that you could find a clone size distribution which is qualitatively very different from what the model predicts. And it's not something that you could fix with matching parameters and things like that. So it could work, it could not work. It is a falsifiable thing. But the tricky area is the area in the middle where it works to some extent. It is not complete nonsense that comes out. It is also not perfect. This is the general situation we find ourselves in modeling biological systems. So then you try to systematically think about the ingredients without just putting your modeling hat on, but also in talking to your experimental colleagues, because things that you do have to make sense. See what could be responsible for the deviation between the theory and the experiment. This is the tedious cycle that we have to go through, whether in physics, biology, any discipline.
That's correct. But now, so here we have this model. Right now we're in a phase of the science that we actually try to understand how this system works at all. Right? This is also what you made clear in your talk. There's a lot of aspects of this immune system and its responses that we just don't understand. But the next step would be to control this immune system. So imagine we move through this phase. Now you have the model. We understand how it works. Understanding would imply we can control it. So what kind of control would you envision we should pursue there?
Well, I have a very simple but clear vision on that. I don't know whether it's realistic, but this is what I'm heading for. And that is, in the immune system, there's a very clear division of objects that immune cells can sense, being categorized either as self or non-self, as enemy or friend. And we know one thing, and that is the immune system is capable of changing its mind from one day to the next. And this happens certain diseases when all of a sudden finds that the immune system starts attacking a particular type of cell. And this is not desirable, but we know that the immune system can do it. What we do not know is what actually made that change. How came that change about? So we first need to understand this. we need to understand what is happening when someone develops alopecia, where all of a sudden your hair falls out. Because it's a mechanism that is possible, but it's a mechanism that we would like to exploit and control. Because if I can manipulate this, if I can tell the immune system which objects are from today onwards to be seen as enemy, which until now was seen as friend, then we can redirect the immune system to attack anything we like.
Mm-hmm.
And this is what I see as really the future of medical applications of the immune system. And this can be especially relevant in cancer medicine, where there are different levels of ambition one can have. So one level of ambition I think is a bit too far for now, and that is to train the immune system to see the difference between a healthy cell and a cancerous cell of a given organ.
Mm.
But we don't need to do that. We can also lower our ambitions and say, look, there are certain organs that a person can live without. Yeah, thyroid is one example. If we could at least instruct the immune system to regard all thyroid cells as enemies, then you could treat the patient first with surgery, remove the thyroid, and then you switch your immune system to attacking all remaining residual thyroid cells. And the difference between that and the normal route, which is chemotherapy, is that if you do it right, you will not have any off-target pathologies that you create, any damage that you create. And it is an active agent. It is actually, it is not a passive chemical. It is an active agent that will seek out anything that looks like a thyroid cell.
Mm-hmm.
So I can see this as the future.
But isn't it, there's some molecular stuff going on there which is not captured in your model yet, presumably?
No, these models are far from that stage.
Yeah.
But I'm now looking ahead. This is one of the things that could become possible once we have an acceptable level of description of the immune system. Because I think we have to be extremely careful at the same time, simply because the immune system is so powerful, and we've seen that in some trials that went dreadfully wrong. Tinker with it the wrong way, you can do tremendous damage too. And
it occurred to me that that's another use for your model, because you can test interventions in the modeling environment before you try them. Because you explained how this particular disastrous trial started out with the successful animal models. So the animal models don't always predict what will happen in a human. But maybe a computational model will show you a variety of possible results, and you could then look out for bad outcomes that you might not have seen in the animal.
Yeah, a colleague of mine from Brazil, from Berlin, once described it in the following way, which I think is a very good way of seeing it. If we build a new car and we crash test it, we don't run Mercedes against walls. We do it on a computer first. That's why we do the crash tests. In medicine, we're not often doing that, sometimes because we don't have the computational ability or we don't have the knowledge to do it. But I think that is the future, exactly as you say. If you have a decent model, and you have to be careful that you test your model against experimental data. But once you get to the stage where you trust your model to some extent, serious extent, then you can crash test ideas. And in the model, you can make those changes that you think on the basis of the theory would be the changes required, and then test whether it actually works like that or does have perhaps unforeseen consequences that are dangerous to the patient.
I think it's more than crash testing as well. I mean, the driverless car research is now moving into simulation because they can't capture enough real-world data of unlikely things that will happen when you have millions of driverless cars. So you want to simulate those situations.
Yeah.
But then there are already attempts underway, as you also mentioned, also failed attempts, to try to really master and control the immune system for more personalized healthcare.
Yes.
So to what extent do you see these current steps as being consistent with the concept of the immune system that you are advancing with this model?
I think it is going in the right direction. In early immunological therapy ideas, very often what was done was non-cell-specific interventions in the immune system, which you can sort of see as similar to just rebooting it, giving it a big kick, and then hope that it settles into a new state, which is doing what it was designed to do. And more recently, especially with this example of these CAR T cells, the research has moved away in the direction of making genetic modifications that are not generic, but they are tailored to the specific needs of an individual patient. And in the case of CAR T cells, they create these receptors to be specifically tailored to recognizing and docking to the cancer cell. Now that is moving already in the right direction. The only thing is it's fairly limited because this only is done for cytotoxic T cells. Mm-hmm. So this doesn't exploit at all the interaction, the network concept, the interaction between the T clones and the B clones. And in that sense, it is still very different. It's not this idea that I have of controlling the self-non-self boundary. That's not what's happening. But at least it is moving in the right direction. It is trying to make non-generic changes. But it's trying to make changes to components that are really tailored to the specific needs of an individual. And they are spectacular, some of these results. It isn't doing everything we would want these things to do. The CAR T cells don't work for solid tumors at the moment. But there's a lot of research going on in seeing what is the reason why it doesn't work there and what can we change. And there have been already several generations compared to the first generation of these methods. They've come a long way. And that's why they've now been approved by the various authorities.
Right. But why would this work by manipulating the T cells and not the B cells? Because you could imagine that the B cells are the ones labeling whatever object you would like to classify as non-self. So can you explain from the perspective of your model why it has been effective on the T cells and not the B cells?
That's a very tricky question because I don't really know the answer to that, and I can only speculate as to why that may be.
But given the model, you must have an intuition, no?
Well, let's approach it from the other side. Would it be a viable alternative to do that? I think it would be. But I need to look very carefully at the differences, if any, between the response of the immune system and the way it does its business in attacking foreign antigen compared to how it deals with deranged own cells. And I would not be surprised if, let's say, the players responsible for the immune response would not be the same. Because B cells are being trained very specifically in the germinal center to look away from the self cells, to really look only at foreign antigen. So it may well be that the way the immune system handles cancerous cells is different, distinct from the way it is most effective in handling foreign antigen. There must be studies looking at that, but I'm not aware of them at the moment. But it is a very good point.
So you moved from theoretical physics to neural networks to the immune system. Do you in any way miss the study of neural networks and the brain, or not that much? You find this more challenging?
That's an interesting question again. And I've had a really good time studying neural networks, but I also find very often that when I've worked on something for 10 years or more, I tend to look for something different. And it can be that different in terms of the modeling and the analysis can be found within the same domain or outside. And what I've seen at the time is that I found all kinds of new things to work on, which happened not to be neural networks. But now, just by accident, I'm moving back and I'm touching that field again, simply because the technology leads me there. I've always enjoyed myself. I thought it was a fascinating area to work in. But at some point, I got the impression that in this community, it became an inappropriate norm that one would have to know everything about biology, everything about the computer science aspects, everything about the theory in order to be effective. I believe that's a fallacy. And I think it has done some damage to that field, and it has sort of pushed communities apart, which I find these things very interesting because I've now, about 10 years ago, moved into what I call quantitative medicine, did a lot of work on protein interaction networks, gene regulation. And I've asked myself, how can we avoid that in the future? So what we've done is we've set up a mechanism where young people are given a program a training program in which they are being taught by people from different disciplines at a level that they can understand, and where we try to educate them to work differently, to not believe that in working in an interdisciplinary team, one has to know everything about all the disciplines, because one ends up being mediocre at everything. But they have to identify what they are in terms of their primary discipline and make sure that they are really on the ball in that. But they need to speak the jargon of the people from at the other side of the various fences, and they need to know what keeps them awake at night. And that's the only crucial thing. But they should not make the mistake to think that they have to be experts at 3 different disciplines. It's not realistic. It's counterproductive. And I think at the time, but maybe I'm wrong, because you know that community better than I do, there was a bit of that happening in between the 1990s and 2000s.
It's still like that.
Yeah.
Yeah. I mean, the multidisciplinarity is clearly still a challenge, also in the field of neuroscience, where there's from the side of the people doing the experimental work, which is extremely intense. Of course, they would feel like, well, these guys haven't made the miles to really appreciate the complexities of what we're doing here. They will not really appreciate the meaning of this kind of data. And the modelers or the theoreticians would often have the feeling like, well, we're not being listened to, right? So there's this massive disconnect. And that's also why I think in neuroscience, theory overall has made less of an impact than you might have wished. But now, okay, given this long trajectory you've made, what should be Ton's law if we want to understand the immune system, the brain, living systems? So what's Ton's law?
Well, I don't think I have one law. I've learned a couple of things, and I can go through them if you want. The first is interdisciplinary interactions, what I just explained to you. And I think where we should move to is a way of working based on respect of expertise on the other side. Right? I mean, if I work with an experimentalist, a bioinformatician, I have to trust them to know what they do, and I leave them to it. What I do need to know is what is it they really find interesting. But what I should not try to do is to convince them what they should find interesting is what I find interesting. Yeah. So these are the types of psychological things I've learned.
That means empathy.
Not that empathy. I just need to know what excites you, Paul. I need to know what is it that makes you really happy. You need to know what makes me happy, and we have to accept that these are different things. But they could all be found in the same project. But they can all be found in the same project. And that's how we can work together best. And I should not try to do someone else's work or even attempt to know what they know. This is completely counterproductive. So that's one of the first things. Generally, I think we live in a bit of a strange time in terms of how science is organized generally. And I think that there will come a point where there will be major revisions. One of the crucial lessons is never sacrifice quality. Everything else you can sacrifice, but never sacrifice the quality of what you do. Be your own worst referee. Yeah. Because fashions will come and go, reputations will come and go, but you want your name to be a sign of quality. And the only way to protect that is to never sacrifice quality. And if you then perhaps are 6 months later in submission of a paper and it doesn't end up in Nature, who cares, right? But maybe in 15 years from now, they will still read that paper. And all the Nature papers will have been seen and forgotten about in 6 months. Quality is everything in science. That should never be sacrificed.
Okay. Another laws you want to add to that, or 2 is enough for now?
No, I drink lots of coffee, Paul.
Okay, good. But so to finish up, so we're going to send Tony from Sheffield to London 5 years from now. Okay. Given the current political situation, let's just assume it's London, might be somewhere else. You never know with Brexit hanging in the air. But okay, there comes Tony. He visits you at King's College to check whether a specific hypothesis was falsified or verified. And that is the hypothesis that you're going to specify today. So what's the one hypothesis that you really would like to see tested in this 5-year frame?
That it is possible to instruct the immune system to change which object it sees as self and it sees as non-self. I think that would be a big, big step forward if we can achieve that and actually have experimental validation for it.
Great. Ton Coolen, thank you very much for this conversation.
Thank you.
Thank you very much.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.